Hello, good afternoon to everybody. The next speaker is Professor Adam Reitzel, with the talk Untangling the Microbiome and its Potential Functions in the cnidarian Nematostella vectensis. Professor Reitzel is a professor of biological sciences at the University of North Carolina at Charlotte. Professor Reitzel is broadly interested in using integrative approaches to better understand the evolution and ecology of coastal invertebrates. Please, I would like to remind you that you can ask questions in the chat and I will read them at the end of the presentation. So welcome, Professor Heitzel. You may start whenever you want.
Thank you very much for the introduction. Let me get my screen shared here. Okay, hey everybody. Yeah, first I'd like to thank the organizers for putting together this excellent meeting. I've really enjoyed listening to all the great research, and also thanks for the opportunity that we will share a little bit of our work on trying to understand aspects of the microbiome of this little sea anemone, which I'll tell you much more about here shortly. But before I get to the data, it's always, of course, most important to talk about the people that are generating the data. And this is my current lab group here. I'd say we're broadly interested in sort of really evolutionary ecology questions, and we apply a sort of a side of omics or a particular omics techniques in order to understand in greater detail, whether it's the mechanism or whether the history of why does individuals vary in their microbiome, and what are some of the mechanisms involved in what we think are involved in mediating that microbiome. And so I'll introduce you to the researchers that contributed to the data I'll talk about today as I get through it. And so over the last couple of decades, the appreciation for the associated bacteria, unicellular eukaryotes, and other microbes as part of animals, as part of a holobiome, has been increasingly cataloged and appreciated. And much of that has to do with not only studying more organisms, but also greater technologies. And so these associated bacteria, which I'd refer to as a symbiosis because they're living with each other, has all sorts of aspects from mutualism to commensalism to indeed being parasites, what we heard from Dr. Lowe in the last talk. And so we can catalog these in lots of different ways in lots of different organisms. And we happen to study a lot of marine invertebrates as part of our study organisms to try to get at this idea of what is the microbiome and why does it vary? And so if I had a question related to this research that keeps me up at night, it's these types of questions. How, when, where, and why do these microbiomes vary between individuals, between species, et cetera? And so we can think about this. It's a very complex question, of course, once you start getting into it, from looking at that you have a diverse community of microbes, and there's also some viruses in here. I understand that's not part of the proper microbiome, but it's important for the latter part of the talk. They're living in a variable environment. These are estuarine environments, as I'll talk about later, which differ in about any measurable abiotic as well as biotic characteristic. And we happen to be focused on these marine invertebrates. So a sea anemone, which I'll tell you about for most of this talk, Nematostella vectensis. But a lot of the work in my lab over the last couple of years is also focused on the microbiome of sea urchins and other echinoderms. And that's been led by two wonderful graduate students, a former graduate student, Tyler Carrier, who's now a postdoc in Germany, and a current PhD student, Remy Ketchum, who's preparing to finish her PhD. And so we're also interested not only in the adult forms, but also in the embryo forms and larval. I won't have a chance to talk about those much today, but we really take this, what we hope is a very broad look at the microbiome and why it varies. So a lot of the work I'm going to talk about today is a collaborative project funded through the Human Frontier Science Program with Sebastian Frun in Dusseldorf. And the two primary students that initially contributed toward the data you're going to see today were a PhD student, Brittany Phippen, and a former PhD student, Whitney Leach, both of which have moved on to postdocs since this project was initially started. And so if our sort of categorize our worldview over the last couple of years as part of this project and sort of continuing today, it's to try to look at this organism, Nematostella vectensis. And so why did we choose this organism? Well, it has a couple features that we think are quite useful, not only for understanding microbiomes, but also more broadly why sort of labs around the world have adopted it as sort of a model system for understanding cnidarian biology. And so some of the original focuses were of using cnidarians broadly speaking was that they're a member of the phylum Cnidaria, which is a sister group to the bilaterians, which are the sort of the classic traditional model systems, sort of specifically when we look at developmental biology as well as molecular biology. They're super easy to use in the lab. So for someone like me who has a hard time growing houseplants, they're even easier than that. And so we can culture the full life cycle of these organisms in glass bowls in the laboratory. And in those, we can recapitulate all major life history characteristics related to development, including sexual reproduction, which occurs in the laboratory every week, every Tuesday. Asexual, so they clone themselves through a variety of different mechanisms. And in addition, we can bisect them and they can regenerate themselves. And so we can generate expansive clonal lines of genetically identical individuals, which helps us to kind of freeze the genotype, if you will, when we're trying to understand biological questions. They're also useful, and we'll come back to this towards the end. They were the first cnidarian, and indeed the first non-bilaterian animal to have a sequenced genome. It was published about 14 years ago, but it just feels like yesterday. In addition, over the last about 15 years, there's been extensive molecular tools that have been developed as part of a worldwide sort of research community to sort of build up this organism. And then last but not least is the ecology. And this is what really spoke to my heart when I first started working with this organism, is they reside in estuaries throughout most of the continental United States and parts of Canada. They've also been introduced to England as well as Brazil over the last century. And so we take that sort of suite of tools and approaches and ease of laboratory culture, and then we apply that to this one particular set of questions, which is looking at the microbiome. And so we've looked at this from a perspective of population genetics genomics. That'll be part of the talk I'll talk about today. We've done a tremendous amount of work looking at the diversity of bacteria that are associated. There's hundreds of OTUs or hundreds of bacteria that are associated with them in these marshes, and we have many of them under culture in the laboratory. We studied them in the field. In fact, in about 6 hours, I'm leaving to go out to the field tonight to do my first field work after the last year and a half under COVID. And then finally, a lot of molecular biology and gene expression, which I'll also talk about today. So because I can't talk about everything, I'm going to talk about 2 projects, but I did want to give a little context for what have we been working on and what do we know so far. And much of what we've learned about Nematostella's microbiome mirrors the microbiome studies in most other animals and most other plants. And so here's just sort of some of the bullet list is the work that we've done, both published and work we're working on publication now, is that associated bacteria differ by location within their geographic range, as well as time of year. So there's a lot of variation throughout space and time, that the microbiome shifts dramatically during the life cycle. And these are really particularly large transition events at metamorphosis as well as reproductive maturity. So going from an immature juvenile to a mature adult. That the microbiome is highly responsive to the environment in which the animal and the bacteria are living in. So things like temperature, salinity, oxygen, et cetera, impact the diversity as well as composition of the microbiome. That a subset of bacteria fluctuate daily. And so these are very consistent with a circadian clock regulated pattern, just like it is in the human gut microbiome, for example. We see similar patterns with particular OTUs that cycle on a daily cycle. And then lastly, this has been a lot of the work from a current PhD student, Quentin Kruger, who you'll see here in a second, is that the bacterial tolerance is very similar to the anemone's tolerance. And so although we find these hundreds of OTUs, he's growing them and testing their tolerance, it's quite similar to the host. So we don't think it's so much the environmental filtering is heavy, but we think it's something about the interaction of the bacteria and the host together, and maybe the bacteria interacting with the bacteria. And so the first of 2 experiments I want to tell you about today is to look at the impact of gene expression based on exposing anemones to individual isolates from their natural microbiome in 2 different contexts. And so this is the work of Quentin Kruger up here on the left, and then Brittany Phippen on the right. And so the 3 bacteria we chose, these are from isolates from anemones from the field. We chose a species of Vibrio, a species of Bacillus as a gram-positive, and then a species of Pseudoralteromonas. And these were chosen not so much for any particular reason, besides that they are part of the natural microbial community, and they represent 2 gram-negatives and gram-positives. And so we set up these experiments to expose anemones to both live cells that were grown to log phase, as well as then we spun the cells out and we exposed them to the cell-free supernatant, or CFS, to try to see if there's a differentiation in the gene expression by the anemone in response to either the bacteria or into the type of signal it's coming from the bacteria. Is it the cell itself, or is it from the cell-free supernatant that's sort of extruded into the media, so to speak? And so furthermore, just to complicate our lives even more, we did that experiment then in anemones with and without their native microbial community. So we spent about a year developing an antibiotic protocol where we can wash away all of the culturable bacteria, at least, by using a cocktail of antibiotics. We did the gene expression data. It has no real effect after removing the antibiotic. And so then we exposed each of those pools of anemones, whether they had their native microbiota on the left, or we had removed it with an antibiotic cocktail on the right. We exposed them to those 3 bacteria, either live cells or CFS. We did a 24-hour exposure. Then we did RNA extraction and a modified RNA-seq protocol called tag-seq, which is 3' N sequencing to do quantitative transcriptomics. And these data were analyzed with DE-seq and then Go enrichment, et cetera. Okay. And so I just want to kind of give a little flavor about what's going on. This is not a 3-hour talk. And so the idea was, I just want to show that, can the anemone tell the difference between the bacteria? I guess, does it make a difference? And it most certainly does. And so here I'm just showing some plots of how many differentially expressed genes. So these were using a log2 of 1.5 and a FDR of 0.05. And we can see that the number of differentially expressed genes changes or is dramatically different between if antibiotic was used or not. So these are those with their native microbial community. We see a huge number of differentially expressed genes, more than 1,500, a majority of which are actually down expressed whenever they're exposed, in this case, the Pseudoalteromonas species. Whereas if we remove the native microbial community, we see many fewer differentially expressed genes and many fewer in those downregulated. Now, if we compare that from the Pseudoalteromonas now with the Bacillus, we see an exact opposite pattern, which is that the removal of the microbial community with the antibiotic before the addition of the live cells, now we get a flip-flopping of the gene expression patterns. And we'll get into the genes here in a little bit. And then finally, Vibrio had very little effect in many ways comparatively, and we certainly didn't see that huge effect of these sort of hundreds and hundreds of differentially expressed genes with or without the antibiotic. And so of many, many comparisons, here's just one of what that looks like. Are all those differentially expressed genes in any way shared genes across those particular treatments? And in many cases, they really were not. It was sort of it was really in a case-by-case basis. So if we look here on the left-hand side here, we have the 3 live bacteria. So we see that huge different number of differentially expressed genes with the live Pseudoalteromonas with the native microbial community. And only about 81 of those genes were sort of conserved and being part of that differentially expressed thing. Whereas for the Vibrio treatment, although there were differentially expressed genes, none of those were shared with the other 2 treatments. And then similarly, we see a very parallel situation whenever we looked at after the native microbial community had been removed, where again, we see some shared genes, but a lot of treatment-specific differential gene expression. And now what if we compare that then with the live bacteria with the cell-free supernatant? And here, again, I'm just showing the number of differentially expressed genes so we get an idea of the magnitude of how much transcriptional variation was occurring. We see that huge spike here of the live cells with still containing their native microbial community. And whenever we compare that then to the CFS treatment, again, everything else is the same here, except for we're using CFS versus live cells. We then see a really large change where we lose a lot of those downregulated genes. Similarly, well, I shouldn't say similarly, conversely, we see with, whenever we use this Bacillus species, we see just a completely different effect, where now in case where whether there was the native microbial community was present with CFS or was absent, we still see a huge number of differentially expressed genes whenever there was Bacillus was used in all cases, except for using live cells with the native microbial community. And so similar now we can look at those Bacillus genes and look at how many of them potentially overlap between the 2. And we find here that again, a fairly small proportion of genes are sort of present in each of those differential gene expression data sets, comparisons, pairwise comparisons. And we compare that with an LPS treatment. I'm not going to get into it, but LPS is used as a common immune stimulator, but yet it has, of course, a very unique signature, which is unsurprising for a gram-positive bacteria, of course. but still. Similar then, if we look at those now, whenever the native microbial community was removed, now we see a much larger proportion of genes that were shared differential expression, regardless of whether it was live or CFS. And so there's something then about the interaction between not only the host and the particular bacteria or the bacterial signal, but also whether the native microbial community is present. And so you may be asking, well, like what genes are there? And so two things that popped out in Go analysis were looking at the immune system. And so it's been published over the years that this is by Tom Gilmore's group that many species of cnidarians have many of the conserved members of the classic toll-like receptor NF-kappa B pathway, as well as multiple genes in the innate immune system related to the complement system. However, whenever we looked at the gene expression, none of these, or very few, were ever differentially expressed, despite the fact we're using these bacterial sort of species or signals. Instead, what came out of the Go analysis and later looking at the genes differentially was actually what's termed the antiviral pathway, which is really a nucleic acid sensing pathway intracellularly, which involves a whole host of genes here, OS, RNase L. But the one we're going to focus on for a little bit here is this MDA5 RIG-I pathway, which interacts with MAVs, mitochondrial antiviral protein, and then the interferon pathway. And then whenever we looked in the sea anemone, these genes are all present. Except for the interferon pathway is a little controversial. But for these, we show that they are differentially expressed, specifically in the Pseudomonas and the Bacillus. Most of the time, they're upregulated in those cases, regardless of whether it's the live cells or the CFS. Whereas in Vibrio, it appears to be it's almost kind of absent. We see no differential expression of these particular nucleic acid sensors. And so this has us kind of excited because now we think that, and in fact, these are among the most differentially expressed genes in the whole transcriptome. So we have all 20-something thousand genes. These pop out as first, second, or third most differentially expressed genes, usually with a log fold or a log 2 change of about 10. So hugely differentially expressed. And so that got us thinking then that, okay, well, we have some genes that are differentially expressed, but is there evidence then that something like this could potentially evolve? Is there evidence from populations of the species throughout its range? And so we got our first evidence. This is a paper that was published a few years ago with Sebastian's group, where we're looking at do anemones from different locations and thus different genetic backgrounds. We know they're highly genetically structured throughout their native range. And so we just collected anemones from 5 different locations. This was an unplanned experiment in some ways, cultured in the lab for 10 years under Common Garden. And then we used 16S to compare the microbial communities. So these are the 5 locations from Maryland in sort of the central Atlantic coast up through Nova Scotia and Canada. And then we wanted to look at the microbial diversity and abundance of adults, and then we wanted to compare that between sites. And to make a long story short, this is how it came out. And so the middle plot here is then those 5 different locations. Sorry, I've lost my cursor. I know you're here somewhere. Anyway, it's not showing up. But that we found that the microbial community, despite the fact they've been cultured under Common Garden, fed the same way, same salinity of water, et cetera, they still retain identifiably different microbial communities that were most similar based on the geographic origin of the location. And so that's kind of saying, well, then maybe there's something in the genome. And so these anemones occupy, as I mentioned, the Atlantic coast of North America, the native range from Florida up through Nova Scotia. And so we wanted to look at, well, is there evidence in the genomes that may indicate that they could potentially differentially select for different microbial communities? And so this work's been led by a research associate in the lab, Dr. Ed Smith. And to remind you, the version 1 of the genome came out in 2007. It was highly fragmented, of course. It was done by Sanger sequencing. And then a V2 genome is unpublished, but is available within our research community just this year. And this original isolate came from Maryland, so sort of latitudinal intermediate for their geographic range. And so what Ed wanted to look at was, how does the genome vary for distantly related individuals? And this is geographically distant, tracks very well with genetic distance, it turns out. And it could give us insights into the microbiome. And so what Ed's done is that he sequenced and assembled genomes from individuals from 4 additional locations. I just want to tell you a little bit about the data because it's super exciting. And so the way these were, so we have isolates from Nova Scotia in Florida, so sort of the north and south, or sort of high and low latitudes. These were done with a with a PacBio with a tremendous amount of data, hundreds of fold coverage. And then Ed took advantage of some mid-ion sequencing to do some very, fairly low coverage for an individual from North Carolina and an individual from Maine. So they're all long read sequencers, really nice read links and really good assemblies. And so just to show you that the assemblies are pretty good, this is the chromosomal level assembly for the current version. So it's about 15 chromosomes, about 250 megabases. And each chromosome is about equal in size, about 16. And so these are Ed's genomes with the PacBio. They're really solid. In fact, the BOOSCO scores, et cetera, are better, although they're not quite as contiguous. Whenever we compare those assemblies to this new reference, which is chromosome level, which is on the right here, the assemblies Ed has put together, this is for just one of those two individuals, matches up really, really well. So these are very contiguous assemblies, and they align with each other quite well. And so there's all sorts of genomic variation, of course, whenever you're comparing individual genomes, SNPs, and all sorts of structural variants, which are things like duplications, inversions, translocations. But the one I'm going to talk about in our last couple of minutes is copy number variation and its context within the larger set of structural variants. And so if we just look at one sort of snapshot that Ed found when looking for structural variants, this is comparing the Florida individual against that reference genome from Maryland. And there are just a tremendous number of structural variants, and structural variants being things about 50 base pairs and larger. And although many of these are small, i.e. they're sort of centered here, right, on the variant size, some of these are quite large. And whether that's due to an insertion deletion events, repeat expansions, contractions, and then tandem expansion contractions. And while many of these are kind of small, hundreds to thousands of bases, some of these are huge megabases or multiple megabases in size. And so what we want to look at in the context of, can we find anything predictive in the transcriptome between individuals that may underlie some differences in potentially the microbiome, was to look at copy number variants, CNVs. And so these are the ratio then of copy number variants between two individuals, the Nova Scotia and the Florida individual. And you see that many of them are sort of centered right on the zero. So there's no difference, right, in the ratio. It's essentially a 1-to-1. But you have some on these tails here, and there were dozens of them that really are way out on the tails, the distribution for copy number variants that are overrepresented or underrepresented in a particular genome. And one of those that really popped out initially was this one called TRIM56, which showed huge copy number variation from a single copy in the highest latitude to up to 15 copies of this gene. So we have a number of paralogs that are site specific. And so what is TRIM56 and why did we get all jazzed about it? Well, TRIM56 is involved in mediating the monoubiquitination of a particular protein called cGAS, which is a prominent player in the innate immune system related to detecting viruses and eliciting antiviral signaling that works with STING, which is also part of the pathway. And so is it TRIM56 or is everything gone expansion within that particular pathway? And it turns out it's primarily that TRIM is really expanding tremendously. We've also found a number of other genes unrelated to immunity. But whereas cGAS and STING have roughly similar numbers of the kind of the 2-ish copies, with the exception of cGAS in Florida, where there's 4. So it does appear to be that specific to this particular gene, this particular TRIM gene. And so that has us now really excited about identifying now not only these particular genes with copy number variants, but now looking cross genome to identify targets of selection that could be related to differences in the microbiome and at least potentially the virome. And this is part of a newly started project with Dr. Yehu Moran, who's at Hebrew University in Jerusalem, funded by the NSF-BSF program. We just started a couple of weeks ago, where we're going to now be looking at the evolution of these pathways and the evidence of natural selection and the antiviral signaling within the species throughout the Atlantic coast. And if you're interested in postdocing, if you're interested in this kind of work, please reach out to me. And last but not least, I'd like to finish up again with acknowledging all the people that are doing the work. I'm just honored to work with such tremendously energetic and bright scientists in my laboratory group, as well as across UNC Charlotte as part of the Bioinformatics Research Center, as well as I'd like to acknowledge our current international collaborators, Sebastian and Yehu, and our funding from NIH, NSF, BSF, and Human Frontiers. And with that, I'd be happy to take any questions.
Thank you very much, Professor Adam. Your talk was exquisite. I particularly find this subject quite amusing. Um, we have some questions right now. We have a question from Lara Bacaro. She asked, only geographic distance were found to influence microbial communities, or any other extrinsic factor may be playing a role?
Oh yeah, for sure. And so we've looked at, I don't know if I should be sharing my screen or, well, anyway. Um, yeah, so we know, for example, that all sorts of abiotic factors are influencing the microbial community. What we don't know is if sort of the reaction norm or the sort of slope of that reaction differs between individuals with different genetic backgrounds. And so we're trying to get at that now with doing field experiments where we're taking individuals with different genetic backgrounds, transplanting them into the field in a safe way. Don't worry, they're not going to get away. In order to track how it is that different individuals with different backgrounds both can retain as well as recruit different microbial communities based on both a natural environment and pairing that then with the laboratory. Hopefully in 10 years or so, we'll actually be able to do sort of proper crossing experiments, but we want to know more about the regions that might be likely important in the genome before we start sort of doing those family crosses.
We have another question from Wei Song. He asked, what are some known viruses that target Nematostella?
None. That's an easy question. There's no known viruses, not only within Nematostella, but in fact within any cnidarian species. There's been some metagenomic work and metatranscriptomic, which has suggested that corals, for example, may have a herpes-like virus and a few others. It turns out that none of them are culturable at this point, and so none have been isolated. And so what we and others are relying on is correlative data to basically use really deep sequencing to identify viruses based on sequence. And so, yeah, so that's kind of a stay tuned.
Do you have any more questions? You can ask on chat below. Okay, Professor, I think that's that. Thank you very much for
Thank
your
you. Have a great day.
presentation. Thank you everybody for attending.